Every week along the Grand Strand, oyster shells are getting a second life. Students at Georgetown Technical College are partnering with the South Carolina Department of Natural Resources program to recycle shells and rebuild reefs across the coast.
The shells act as a natural filter to get toxins out of the ecosystem.
In a coordinated effort spanning more than a dozen Gulf Coast sites, Florida conservation agencies and NOAA partners deployed hundreds of thousands of tons of oyster shell into degraded estuary waters. And what colonized the seafloor in the weeks that followed stunned the marine biologists monitoring it. That image, tons of ancient shell raining into the dark water below, was only the beginning of what scientists are now calling one of the most dramatic ecological reversals ever recorded on the American Gulf Coast. Florida didn't just dump 500,000 tons of oyster shells into the Gulf of Mexico. They watched an entire ecosystem grow back from nothing. Not slowly, not partially, rapidly, and with a biodiversity that stopped the monitoring teams cold. Researchers from the Sanibel Captiva Conservation Foundation, the Florida Department of Environmental Protection, the Nature Conservancy, and NOAA's Office of Habitat Conservation have spent years tracking what happens when you give a dead seafloor an enormous pile of calcium carbonate and step back. What they found challenges everything the scientific community believed about how long ocean recovery takes. Because what formed on that seafloor was not just a reef, it was a world with hundreds of species, with sharks and grouper and snook appearing within weeks of deployment. A world growing on a foundation of shells pulled from quarries where they had been buried for tens of thousands of years. And the deeper scientists look at what's happening, the more unsettling the larger question becomes. What have we been doing to the Gulf for the past century? And do we have any idea of the true scale of what we destroyed? That discovery was only the beginning. What came next changed everything. To understand what happened offshore, you need to understand what was there before, or rather, what wasn't there. For most of the past century, Florida's coastal seafloor, particularly in the estuarine systems that fringed the Gulf of Mexico, was, in the words of marine ecologist Ann Birch of the Nature Conservancy, biologically barren. That's not a metaphor. Vast stretches of the Gulf bottom were flat, featureless, soft silt. No vertical structure. No shelter, no surface for anything to attach to, grow on, or hide beneath. The kind of underwater landscape where juvenile fish disappear and don't come back. It was not always this way. Before European settlement, Florida's estuaries were studded with enormous oyster reefs. In Apalachicola Bay alone, a body of water in the Florida Panhandle fed by one of the largest river systems in the Southeast, there were once 10,000 acres of active oyster habitat, reefs that rose from the seafloor in layered, cathedral-like formations, reefs that had been growing, generation by generation, for millennia. A North Carolina study conducted on a single comparable reef documented 303 distinct species living within its structure. 303 species in one reef. The indigenous peoples who lived along Florida's Gulf Coast understood exactly what they had. Archaeological evidence, including peer-reviewed research published in the Journal of Archaeological Science, reveals that woodland period communities on Florida's Gulf Coast practiced what researchers now call mariculture, intentional, multi-generational management of oyster beds. These people were not simply harvesting, they were tending, culling weaker individuals to improve shell density, selectively removing valves to create the hard substrate that the next season's larvae would need to settle, running what amounts to a shell restoration program with Stone Age tools. Then came the industrial era, and then the boom. By the late 1800s, the Apalachicola region alone was supplying 90% of Florida's oysters and 10% of the entire nation's. Oyster houses lined the waterfront. Tonging, culling, and shucking were skills passed from parent to child across generations. Annual harvests in the southeast peaked in 1908 at nearly 20 million pounds. And then, in the span of roughly 1 century, almost everything fell apart. Overharvesting stripped the reefs faster than they could rebuild. Industrial and residential effluent poured nitrogen, chemical contaminants, and sediment into coastal waters. By 1971, a die-off erased more than 90% of commercially harvestable oysters in the Pensacola Bay system. Disease moved into reefs weakened by pollution. Boat traffic scarred reef edges. Coastal development reshaped the hydrology of every major estuary in the state. And then, the Deepwater Horizon oil spill of 2010 delivered a catastrophic blow to an oyster population already in collapse, one from which the Gulf has not recovered. By the time Florida's resource managers began seriously planning large-scale restoration, the numbers were devastating. According to both NOAA and the Nature Conservancy, more than 85% of the world's oyster reefs have been lost over the last 2 centuries. In Apalachicola Bay specifically, the figure is worse. Fewer than 500 acres of healthy oyster habitat remain out of the original 10,000. A 95% collapse. And as of 2023, Florida's commercial oyster harvest had declined 94% in weight and 87% in value from its 2012 peak, the lowest recorded harvest in the state's history. The Gulf seafloor wasn't barren because nothing wanted to live there. It was barren because the architecture was gone. Picture the scene below the water. Pale, featureless silt in every direction. A desert floor extending for miles. A place where a larval oyster, barely visible to the naked eye, barely a grain of sand, has nowhere to land, nowhere to attach, and nowhere to survive. Without a hard surface, the spat drifts, sinks into the silt, and dies. The ocean, in this condition, cannot remember what it was. It has no surface on which to begin. That is the problem that 500,000 tons of oyster shell was placed in the water to solve. The deployments, involving coordinated efforts by the Florida Department of Environmental Protection, NOAA's Office of Habitat Conservation, the Sanibel Captiva Conservation Foundation, Tampa Bay Watch, the Nature Conservancy, and a network of community organizations, were not glamorous operations. Barges loaded with recycled restaurant shells and fossilized shell material mined from Charlotte County quarries crept slowly out into Gulf waters under the punishing Florida sun. The shells had been curing on pallets for months, a minimum of 3 months according to DEP protocol, to allow natural elements and microbial activity to clean any remaining tissue. The smell, by all accounts, was formidable. Water cannons mounted at the stern blasted the material overboard in coordinated arcs, cascades of calcium carbonate splashing down into the green-gray murk below in patterns precisely mapped by the marine biologists on deck. Nobody on board knew exactly what would happen next. They had models, they had theory, they had precedent from smaller test projects, but at this scale, across dozens of sites stretching miles of degraded coastline, the ecological outcome was genuinely unknown. What happened next surprised even the most optimistic scientist in the room. The first sign came faster than anyone expected. When research divers from SCCF's Marine Lab descended to assess a newly deployed shell field in the Caloosahatchee River estuary, a body of water at the mouth of one of Southwest Florida's major river systems, they found something on the fossilized shells that stopped them mid-dive: oyster spat. Not weeks or months after deployment, days. Tiny, translucent juvenile oysters had already located the shells and glued themselves permanently to the calcium carbonate surface using a biological cement that no synthetic adhesive has yet replicated. The attachment is irreversible within hours of contact. Once an oyster settles, it never moves again. It commits entirely. This is the process marine biologists call settlement, and understanding why it happens with such speed is central to understanding everything that followed. Eastern oyster larvae, Crassistria virginica in its juvenile free-swimming stage, spend 2 to 3 weeks drifting in the water column after hatching. During that window, they grow a foot-like appendage and begin probing surfaces with a chemical sensitivity that is still not fully mapped. They are, in a very real sense, reading the ocean, testing substrate after substrate, measuring chemical signals, assessing texture, evaluating pH profiles. And the surface they prefer, above all others, is oyster shell. The rough calcium carbonate of an oyster shell releases molecular cues, signal compounds that have been identified in laboratory settings but whose full biochemical complexity remains an active area of research, that tell a drifting larva it is found in appropriate settlement zone. The texture, the mineral composition, the microscopic topography of the shell surface, all of it is read by the larva as a green light. Once settled and cemented in place, the spat begins to grow, roughly 1 inch per year under optimal conditions. At the Caloosahatchee site, SCCF had placed approximately 3,045 tons of fossilized shell, 6.09 million pounds, sourced from a Charlotte County mine that excavates Pleistocene-era reef deposits, in a carefully coordinated deployment involving barges and a specialized water cannon. Within days of placement, settlement had begun. Within weeks, the species inventory was expanding faster than the monitoring protocols were designed to track. Eric Milbrandt, director of SCCF's Marine Lab, documented species at the new reef that local fishermen and researchers hadn't reliably seen in those waters in years. Bull sharks cruising the structure's perimeter, Goliath grouper, a federally protected species that can exceed 700 pounds, hovering in the shadow of the growing shell field, snook, gray snapper, redfish, and stone crab establishing themselves in the crevices between shells. The numbers, when they came in, were striking. A single square yard of mature oyster reef, per species surveys conducted in Florida's Indian River Lagoon, contains an estimated 5,895 individual oysters, the equivalent of 45 bushels. And that same North Carolina study referenced earlier found 303 distinct species using a single reef as habitat. 303 species on one reef. That figure had seemed almost impossible before the monitoring data from Florida's restoration sites started coming in. Now, it seemed obvious. The ocean had always been ready. It just needed something to land on. Take a moment with that. The seafloor that looked like nothing, flat, featureless, ecologically dead, contained the dormant potential for hundreds of species. All of it waiting, invisible, drifting in the water column for a surface that had been absent for a century. But the physical recovery of the reef was only the most visible layer of what the shells were triggering. Because oysters don't just create structure, they transform the chemistry of everything around them. A single adult eastern oyster filters up to 50 gallons of water every 24 hours. That is the measured, documented filtration rate of one individual oyster, verified under controlled conditions by the Florida Department of Environmental Protection and corroborated across multiple independent research programs. It is not an estimate. It is a function that runs continuously, around the clock. Every day the animal is alive. Scale that capacity across a restored reef. Millions of individuals, packed at densities of nearly 6,000 per square yard. And the numbers enter a different order of magnitude entirely. A 3-acre reef of dense oyster habitat can filter the equivalent of an Olympic swimming pool's worth of water within hours. And what it extracts from that water is not just sediment and algae. Oysters actively remove suspended bacteria, chemical contaminants, phytoplankton, and, critically, nitrogen. Florida's coastal waters have been in a nitrogen crisis for decades. Lawn fertilizers, agricultural runoff, and failing septic systems have leached excess nitrogen into groundwater and surface water at rates that have fundamentally altered coastal chemistry. When nitrogen enters an estuary in concentrations beyond what the ecosystem can absorb, it triggers explosive algal blooms. Florida residents know the consequences intimately. Red tide events that coat beaches in dead fish, toxic blooms that close shellfish harvesting areas for months, oxygen-depleted dead zones where nothing survives. Oysters are one of the very few natural mechanisms capable of countering this cycle at scale. As oysters filter nitrogen-laden water, they incorporate the nitrogen into their tissue and their shells. When the oyster dies, a portion of that nitrogen undergoes denitrification, a bacterial process that converts it into harmless nitrogen gas, releasing it permanently from the water column. The reef doesn't just clean the water, it removes the excess nutrients that would otherwise fuel ecological collapse, and it holds that removal for as long as the shell persists, which, as the Charlotte County fossil record shows, can be tens of thousands of years. Tampa Bay Watch's Sarah Herndon, Habitat Restoration Director for the Shells for Shorelines program launched in 2022, documented measurable improvements in water clarity at monitored reef sites within months of construction. With improved clarity came increased sunlight penetration. With sunlight came seagrass regrowth in adjacent areas. And seagrass brought its own cascade: nursery habitat for juvenile fish, foraging grounds for manatees and sea turtles, a root system that began stabilizing the soft sediment floor that had been eroding for decades. This is what marine ecologists call a positive feedback loop. Each recovered element enables the recovery of the next. Oysters clean the water. Clean water grows seagrass. Seagrass stabilizes sediment. Stable sediment keeps the water clearer. Clearer water grows more seagrass. And through all of it, more species arrive, adding further biological complexity to a system that, left alone, continues to accelerate its own healing. The data was unambiguous. The implications were anything but simple. Because if this process is this powerful, and if the technology to initiate it is genuinely as straightforward as deploying the right shell material in the right place, the question that kept surfacing in every research debrief was the one nobody wanted to say out loud. Why did we let 95% of this disappear? The water held that question for a while. So did the research teams. Here is where the story insists on honesty. Because the shell deployments were not universally successful, and the reasons they weren't are as important as the reasons they were. In 2023, a peer-reviewed study published in Marine and Coastal Fisheries, led by researchers including Dr. William Pine of the University of Florida, examined restoration outcomes across 3 major Florida Panhandle estuaries: Pensacola Bay, St. Andrew Bay, and Apalachicola Bay. The paper's conclusion was blunt. In multiple study sites, oyster populations did not increase as restoration theory predicted they should. The culch, the technical term for shell substrate, was placed correctly. Larvae were presumably present. The model said the wreaths should form. They didn't. Not in those sites. The culprit, the research identified, was salinity, or more precisely, the chronic disruption of the natural salinity regime that eastern oysters evolved within. Chrysostria virginica is exquisitely sensitive to the balance between fresh and salt water. The animal thrives in the 14 to 28 parts per thousand range of brackish salinity typical of a healthy estuary. Below that range, oysters close their shells and stop feeding. Above it, predation and disease accelerate. The narrow band of conditions that allows an oyster reef to function is not negotiable. Biology set that range. It cannot be overridden by shells. In Apalachicola Bay, the salinity has been destabilized for generations. Cities in Georgia draw heavily from the Apalachicola-Chattahoochee-Flint River Basin, the same system that feeds freshwater into Apalachicola Bay. During droughts, insufficient freshwater reaches the estuary, driving salinity too high. When Lake Okeechobee's floodgates are open to manage water levels upstream, a surge of freshwater crashes through and drives salinity too low. The oysters, caught between extremes, cannot establish. Shell alone cannot fix a river. Matt Posner, executive director of the Pensacola and Perdido Bays Estuary Program, has said this clearly. Shell is necessary, but not sufficient. Water quality, freshwater flow, nutrient levels, these are not secondary concerns. They are the preconditions that determine whether any restoration investment succeeds or fails. Skeptics of large-scale shell restoration programs point to exactly this dynamic. You can spend millions on substrate deployment and produce nothing if the underlying hydrology is broken. You can mistake ecological theater for ecological recovery. But here's what the same research confirms. In the sites where those underlying conditions were right, where salinity was stable, where water quality had improved, where freshwater flow was adequately managed, the results were not just positive. They were exceptional, beyond projections, beyond what any prior restoration model had forecast at this speed. Which means the failed sites are not a refutation of the approach. They are a diagnostic. They tell scientists exactly where the effort needs to be redirected, not away from shells, but toward the systemic repairs that allow the shells to do what they're biologically designed to do. The ocean is not asking us to engineer its recovery. It's asking us to stop disrupting it. And it has demonstrated, in site after site where we've managed to do that it knows exactly how to finish the job. If you're finding this as compelling as I am, a like genuinely helps this channel keep investigating these stories. Now step back from the restoration sites. Step back from the water quality data and the species inventories and the salinity curves. And consider what this entire story is actually pointing toward. A species, Crassostrea virginica, the eastern oyster, spent thousands of years building underwater infrastructure along Florida's coast. Structures capable of supporting over 300 species simultaneously. Structures that cleaned the water, stabilized the shoreline, buffered storm surge, sequestered carbon, cycled nitrogen, and fed human communities across centuries. And then, in the span of roughly 100 years, we destroyed 85 to 95% of that infrastructure. Then we took some of the shells, the calcified remains of what had been there, and put them back into the water. And within days, the ocean began to rebuild. Some marine ecologists are now arguing, with increasing conviction and published data to support them, that oyster reef, not coral reefs, are the truly foundational reef ecosystems of the world's temperate coastal zones. Coral reefs get the documentaries, the conservation fundraising, the cultural recognition, but in terms of per-acre ecological productivity, water filtration capacity, fish nursery function, shoreline protection, and direct economic benefit to coastal communities, oyster reefs may be the more critical and the more recoverable of the two. Anne Birch of the Nature Conservancy has called oysters the quiet, unsung heroes of our estuaries. That framing may still undersell what the restoration data is revealing. Because the evidence doesn't suggest that oysters are helpful additions to a healthy estuary, it suggests that a healthy estuary in Florida and along the entire eastern seaboard may be functionally impossible without them. One theory gaining traction in coastal ecology, not yet consensus, but discussed in peer-reviewed literature and at fisheries management conferences, proposes that the widespread collapse of American commercial fish populations over the 20th century may be attributable not solely to overharvesting, but significantly to the parallel and largely untracked collapse of oyster reef nursery habitat. Remove the reefs, and juvenile fish lose their shelter. Without shelter, survival rates among juveniles plummet. Without juvenile survival, adult populations fail to recruit. The fishery collapses, and we blame the nets. Some researchers argue this may be one of the most significant ecological mechanisms driving the decline of American coastal fisheries, and one of the least studied, because the reef loss was so thorough and so early that baseline data barely exists. If we take this framework seriously, then what the Florida restoration projects are rebuilding is not merely oyster habitat. They are rebuilding the foundational nursery architecture on which every other commercially important species in the Gulf depends. The appearance of bull sharks, Goliath grouper, redfish, and snook at new reef sites within weeks of shell deployment is not coincidental. It is these species moving toward the substrate that should have been there for 100 years. There is one more dimension worth raising, one the science has not yet fully worked out, but which the data hints at insistently. What is the carbon accounting of an oyster reef? Oysters build their shells from calcium carbonate, extracted from dissolved inorganic carbon in seawater. As the animal grows and dies, and its shell persists, that carbon is locked in a solid mineral form that can endure, as the Charlotte County fossil record demonstrates, for tens of thousands of years. A large, mature oyster reef is not just a biological structure, it is a geological one, a long-term carbon repository, built by biology and maintained by ecology. At the scale of 500,000 tons of deployed shell, with millions of new oysters growing on top of that substrate, the potential carbon sequestration implications are not trivial. The science on this is still being quantified, but the direction it points seems clear. The ocean built a carbon sink once. We destroyed it. It's waiting to build one again. In Apalachicola, the small panhandle town that once supplied 10% of the entire nation's oysters, there is a man named Roger Mathis who learned to oyster from his father at age 6. He is 68 years old now. He owned an oyster house for 35 years. He drove his product to loyal customers in Tampa and Lakeland. He watched the reefs thin out in the 1980s, accelerate into decline in the 1990s, and collapse entirely in 2013. In 2020, the state of Florida issued a 5-year ban on wild oyster harvesting in Apalachicola Bay, an emergency measure to protect whatever biological remnant remained from further extraction. The ban upended what was left of a generational industry and a way of life that had defined the Forgotten Coast for over a century. He calls himself and the remaining old-timers a dying breed. The Florida Fish and Wildlife Conservation Commission's 2025 staff report confirmed what Roger Mathis already knew in his bones. In 2023, only 119 people commercially harvested oysters in Florida, a greater than 90% collapse in participation from the industry's most recent peak. These are not abstracted economic statistics. They are human beings who no longer have a livelihood, communities that no longer have an identity, a culture that is disappearing faster than the reefs it depended on. In January 2026, the state formally lifted the Apalachicola ban. The recovery it authorized is real, but fragile. Some monitored areas show genuine signs of reef reestablishment. Others remain ecologically depleted. Scientists from Florida State University's Apalachicola Bay System Initiative are still developing the adaptive management framework that will determine whether the next decade of investment produces a functioning bay or another cycle of disappointment. But something is materially different now than it was 5 years ago. The shells are in the water. Across dozens of sites spanning Pensacola Bay, St. Andrew Bay, Apalachicola Bay, Tampa Bay, the Caloosahatchee Estuary, and the St. Lucie Lagoon, the seafloor has changed. It has structure. Oyster spat is finding it. Fish are finding the oysters. Sharks are finding the fish. The Nature Conservancy's 33 reefs along 6.5 miles of Pensacola East Bay shoreline, funded entirely by Deepwater Horizon settlement money and monitored by the National Fish and Wildlife Foundation, are producing oysters. Tampa Bay Watches program is filtering water that hasn't been biologically filtered in living memory. SCCF's Caloosahatchee Reef is drawing Goliath grouper to a river mouth where they haven't reliably appeared in a generation. None of this makes up for what was destroyed. Not the 10,000 acres of Apalachicola Reef, not the century of cultural identity built around those reefs, not the Roger Mathises of the world who spent lifetimes on the water and watched it hollow out around them, while the institutions responsible for managing it looked the other way. But it reframes what is possible, and it raises a question that is simultaneously the most hopeful and the most uncomfortable thing this research suggests. The ecological recovery we're witnessing may not be something humans are achieving. It may be something we're finally, incrementally, stopping from being prevented. Nature has been ready this whole time, waiting in the water column, drifting, looking for something to land on. We gave it shells, and without hesitation, the ocean remembered everything. There are now more active oyster reef restoration projects underway along Florida's coastline than at any point in recorded history. NOAA has committed $5 million through the Bipartisan Infrastructure Law to expand Gulf Coast shell recycling programs across Louisiana, Florida, Alabama, and Texas. Tampa Bay Watch is building reefs at MacDill Air Force Base and Abercrombie Park. The Sanibel Captiva Conservation Foundation continues to pull fossilized shell from Charlotte County mines and return it to the estuary system that produced it tens of thousands of years ago. Florida State University and FWC researchers are testing concrete, Kentucky limestone, and experimental substrate configurations in Apalachicola in the hope that some combination of materials and placement strategy and freshwater management can compress what would otherwise be a decades-long recovery. And here is what the data has established beyond reasonable dispute. Where the underlying conditions support it, the reef comes back, faster than models predicted, with greater species diversity than baseline surveys suggested was possible. Self-seeding, self-expanding, self-sustaining once it crosses a critical threshold of biological density, requiring no further human input to keep going. The eastern oyster does not need our engineering to build a reef. It never needed our help. What it needed, what it still needs, is for us to stop actively preventing what it has spent thousands of years perfecting. So here is the question the scientists are sitting with. Not a rhetorical one, a genuine one, with genuine implications for every coastal ecosystem in the developed world. If deploying 500,000 tons of shell produces results this striking, this quickly, across this many species, what happens when restoration reaches the scale of the original loss? What comes back when we return not a fraction of what was taken, but all of it?